Can we utilize personalized medicine to prevent type 1 diabetes? Mike is professor in chief of pediatric endocrinology at the University of Florida. He's been involved in type 1 diabetes research since the early 1900s, no, the 1990s. He trained he got a master's of clinical investigation at the University of Florida, as well as his MD, and before that, a BS from Duke. His research focuses on predicting, preventing, and ultimately reversing type 1 diabetes. And he participates and leads numerous clinical trials utilizing immunotherapy. Mike is the PI of the University of Florida's TrialNet Clinical Center. He's the chair of the TEDdy Clinical Implementation Committee and the PI of the University of Florida and Stanford ECHO Collaborative. He leads and participates in technology studies seeking to improve the lives of people living with type 1 diabetes using pumps, CGMs, and systems that automate insulin delivery. And we very much look forward to your talk. So welcome, and I wish that we could have taken you to dinner, but your travel was a lot easier.
Thank you, Dr. Goland. I appreciate the opportunity to speak with you, even though it's virtual. I would love to come visit New York, and I'll certainly take a rain check on the dinner sometime. And thanks for that lovely introduction. I'll be sure that the check is in the mail from my mom. She'll be very proud. All right, so let's chat today a little bit about how we can try to move towards personalized medicine to prevent type 1. And really that part's going to be at the end, because that's a little bit more conceptual than I've got details on. But I'm going to share with you a little bit of what we've been working on here at UF and beyond with that goal in mind as it relates to using combination therapies. And of course, I'm really fortunate to work at the University of Florida Diabetes Institute, where we have a long history of collaborative team science. And so coming from the state of Florida, I couldn't help but think about how we do everything translationally, working from mouse models, although usually not named Mickey. And we really try to convert them into things that are going to help our patients at the end of the day. So even the most basic of our science work is almost always focused on these kids that we're trying to help. All right, so just for disclosure purposes, I have funding from NIH and Helmsley Trust. I'm on the board of a company that's working to develop a humanized ATG that, if it works, would eliminate the serum sickness side effect. And I'll be talking a lot about ATG today, although not this particular product. You're welcome to take pictures of anything I show or screenshots or share them. I don't like keeping anything secret, so share away. And you can tweet me if you'd like. All right, so this is the classic model of type 1 that George Eisenbarth blessed us with many years ago. And it still holds really true in so many ways. Namely, patients have a beta cell mass that increases very marginally early in life, but then is relatively stable. Most patients with type 1 have some genetic predisposition, some things happening to them, and then they start this course towards reduced beta cell mass, beta cell function, and ultimately type 1. I think the big changes in this model, which have come more recently, are that the rate of decline is probably a lot more variable, or is a lot more variable in the patients that we see. Anybody taking care of patients has sort of known this always, but it was easier to draw models with a straight line. But the reality is it's just like any other autoimmune disease, in that it's probably waxing and waning, both in the severity of the autoimmune attack and in the loss of beta cell function. We also know that there's a huge variability in when patients are going to get type 1 clinically, as relates to how they're falling down this slope towards having not as much beta cell function left. And then also, really importantly, there's lots and lots of data to support this over the last couple decades. Many patients with type 1, even those who've had type 1 for quite some time, make very meaningful levels of C-peptide. And we should be working to re-enfranchise those folks in clinical trials and immune therapy opportunities to preserve that beta cell function. Because even though it might seem to us historically as being relatively minimal, it does appear from all data that even protecting those small amounts of beta cell function can provide real meaningful long-term benefits to our patients. So, how do we go about that? The system is obviously inherently quite complex, and that's both a good and a bad thing in that it gives us a ton of targets to go after. So, do we focus on antigen-specific therapies? Do we focus on things that are beta cell protective? Should we be looking at pure immunosuppressive agents like anti-CD3 or thymoglobulin, or more immunomodulatory things like alefacept? Do we really look at very specific HLA blockade like methyldopa and DQ8, and on, B cell function, cytokine blockade? Do we use T cell therapies? And I think historically our field has been handicapped by the idea that we, as purists, as scientists, want to study one, regimen, one therapeutic blockade, one pathway at a time. And largely that's resulted in pretty minimal effect, to be very honest. And so we've entered an age which is not all that novel, of asking why we shouldn't be approaching type 1 diabetes like we approach many other diseases, and that is with combination therapies. So I always show this slide just to point out that, Des is one of my main mentors. I've known him since I was a teenager, and Mark Atkinson and Edwin Gale wrote this editorial now almost 20 years ago saying, why can't we prevent type 1, and arguing that we really should be approaching this with combination therapy. And, certainly over the last 20 years, the idea of this has gained a good bit of traction. But for practical purposes, we still are in the infancy of doing things in combinatorial ways. And the concepts for it are pretty straightforward. Not to be a pessimist, but single immunotherapies have in many ways been largely ineffective. So, don't keep doing what doesn't work, as Einstein would have said. Combo therapies should allow us, at least conceptually, to approach therapies that might have historically been thought of as risky, but with lower doses, if we can really create some synergy. We should, now that we understand the biology of the disease much better, be able to come up with more rational combinations that attack different components of the disease, and perhaps thinking towards personalized medicine, different components for specific patients who have different pathways involved in their type 1. Clearly, there are models in other diseases where combination therapy has led the way, namely cancer, HIV, and transplantation. And then, as a practical aspect, somebody who I always think of myself as a clinician who happens to do research, not the other way around. I'm not a patient guy. I'm always wanting things to go into patient care as soon as we have evidence to support them. We have, ample data with agents that already have FDA approval, although typically in other disease processes. But we should be using those agents to try and repurpose them for type 1. And that's something that we've been doing quite a bit in the last decade, and now with some success. So just again, as a historical reminder, the idea of combination therapy is not a new one. This paper from Janet Silverstein is my very first mentor back in the late 80s, when she was still doing immunotherapy trials, showed very nicely that azathioprine on steroids actually provided a pretty impressive response in terms of peak serum C-peptide and glucose response in treated versus control patients. Of course, the problem here was the side effect profile of the approach, and many people were kind of scared away from this, including Janet, who swore that she would never do another immunotherapy study after this one with a drug that was that toxic. But the field has slowly learned from these experiences. I think we have better drugs, and we're wanting to move forward in a way that really helps our patients, and it's still going to require thinking of how to use these things. So again, you have to decide if you want to be an optimist or a pessimist, right? So if you look at some of the major trials done in the last decade, this is how you could look at it. So I think it's important to point out that combination therapies don't always work, and sometimes they're actually bad. While this was a pilot study, the combination of IL-2 and RAPA, again, not really powered to look at beta cell function per se, but certainly it had transient adverse effects on beta cell metabolism. So some combinations might actually not make things better, but might make them worse. A TrialNet study of MMF and DZB didn't work at all. So not worse, but that combo didn't help us. So again, we have tried. And then a bunch of single therapies, which again you could say have either not worked at all or been marginally effective. GAD seemingly doesn't do anything. Although recent data, when you give it directly into the lymph node, may be suggesting that there's some benefit there. Canakinumab did nothing. Tight metabolic control did nothing, despite all the previous evidence of trying to do biostat and put people on maximal insulin right at diagnosis. And then there's a handful of things that have worked, but worked is all, eye of the beholder in terms of how you define worked. When you talk to new onset patients, they're expecting you to say, I've got something that's going to reverse their disease, not need insulin for some significant period of time, if not in their minds forever. And obviously we don't have anything that really offers that to date. That said, we're not striking out entirely anymore. We have this list of drugs that does do something, and I think something meaningful for most of us who've been in the field for a while, appreciating most recently, I think our ability to delay movement towards clinical type 1 with teplizumab and the possible labeling of that drug by the FDA, which would be a huge paradigm shift for us, as well as the effects we've seen with alefacept, unfortunately no longer on the market, but Ritux, abatacept, and anti-thymocyte globulin, which I'll talk about a bit more. All right, so I show this more of as a thought experiment than like what we think we should be doing nowadays, but it is important to just remember how we got here, or at least how our group got to this point. So I would call this the most extreme example of combination therapy in man to date in the type 1 space. In our group, we just call this for short the Brazil approach, because it was led by a doc named Julio Voltarelli down in Brazil. But it actually came from a transplant immunologist at Northwestern, Richard Burt, who tried to get people in the US to do this, and they all just said, you're crazy. Nobody's going to do this kind of therapy in the States. This is way too risky. And that's because it was, essentially a stem cell transplant. They utilized a stem cell mobilization with Cytoxan, okay, bad drug, GCSF, granulocyte colony stimulating factor, and then they harvested the CD34 counts. And then they did what is, I think, arguably non-myeloablative therapy with high-dose Cytoxan, high dose to me, but not as much as you would normally give in true transplant. And that's why they call it non-myeloablative, meaning you should recover regardless of whether or not you need to get cells back, but still enough that people are going to be extremely lymphopenic without some additional therapies. And to induce this myeloablation, they also used anti-thymocyte globulin. And then they did what they called a mini transplant, which is they gave them back the harvested CD34 cells along with some GCSF to stabilize and promote production of new cells. And then they had to give, antibiotic prophylaxis and keep these patients in the hospital for several weeks. They were treated like they were chemo patients. And despite how complex that sounds and frightening, the outcomes are the kind that don't require statistics to say that works, again, in what our patients are kind of thinking. 20 of 23 of the patients who underwent this therapy became insulin-free for at least a month. 12 of them were insulin-free for more than 14 months, with the mean of that group being 31 months insulin-free. Again, you don't need stats for people to say that works. And better yet, it wasn't because they were having poor control. A1Cs were less than 7% in this group. And they didn't have what we always in our field call an effective therapy, and that the C-peptide is declining at a slower rate compared to placebo. They saw a frank increase in C-peptide out to 2 years. Okay, so really, again, some of the most impressive results I think we've ever seen in the type 1 space. But, and a big but, this approach has got some significant short and potential long-term side effect concerns. And so our group really thought, let's try and deconstruct the Brazil approach and see if we can put it back together in a way that's kinder, gentler, and still might have some effect, and be more palatable to US audiences and patients elsewhere. And so we lovingly call that the Brazil Light approach. So let me just review quickly these drugs in case folks in the crowd aren't used to thinking about them. So the first one that we thought we could use, we immediately said we're not using Cytoxan. That was just kind of off the board. But thymoglobulin seemed reasonable. An FDA-approved drug, been around for 25 plus years, most commonly used as a renal transplant drug. And it's a biological agent more than a drug, right? It's a polyclonal mix of rabbit anti-human antibodies made by taking thymus tissue from kids having chest surgery and putting it into these lovely rabbits that Sanofi owns. And it causes a very impressive T cell depletion and a bit of an immune reset. And so we took it to the animal model, where, when I say the royal we, including George Eisenbarth, had done studies in the NOD mouse long ago showing that it provided pretty good reversal in the NOD. But we all know the mouse model is only the mouse model, and not much translates well to humans. George's team had done early human studies and shown there was moderate efficacy with anti-thymocyte globulin, a previous product very similar to it, but they largely abandoned that approach because of side effect concerns, just like we talked about with azathioprine and Dr. Silverstein's efforts. And as we were thinking of this, there was already a monotherapy trial in place looking at ATG at a higher dose. We refer to it high dose, still not high dose compared to what transplant patients get. But the START trial, led by the Immune Tolerance Network and Steve Gittleman's group, was looking at ATG at a dose of 6.5 milligrams per kilogram total dose. And so we thought it made a lot of sense to sort of include anti-thymocyte globulin from the Brazilian effort in our combination approach, but at hopefully even lower doses. The other drug that we were most interested in using from the Brazil study was GCSF, or the trade name is Neulasta for the long-acting pegylated form. And I won't focus on this because it'll become clear in a moment why, but this drug essentially we thought would mobilize and stabilize Tregs and dendritic cells. And we know that it does prevent diabetes in the NOD, but prevention in the NOD is even easier than reversal. And when we tried to do reversal in the NOD, that did not work. There is some data in other autoimmune diseases whereby giving GCSF improves the disease and reduces severity. And so I won't give you any details, but we did do a monotherapy safety trial in new onset type 1s with GCSF so that we could move towards combination approaches. And we showed that it was incredibly safe, but really did nothing beneficial for the patients in terms of their C-peptide production. But perhaps what was the most exciting for us as we were trying to figure out where we go, mouse to man, man to mouse, were these sets of experiments. And this summarizes a number of times that we did this study, is repeated by us and other labs, where we were trying to understand which combinations of these drugs will reverse diabetes in the NOD mouse. And as you can see, you've got percent normal glycemic here on the x-axis and days on the y-axis and days on the x-axis. And in the black, you can see control, they all revert to getting diabetes. We give them an insulin pellet that saves them for a couple of weeks, just like you would treat a patient. But unfortunately, when the pellet runs out, they all revert. GCSF by itself in blue did almost nothing. ATG at a low dose in green reversed about 50% of the animals durably. So it's really impressive, just 2 doses, and then they didn't need it ever again. But the real magic sort of was in these red and yellow lines, which was when we combined GCSF, which again by itself did nothing, with a low dose of ATG, or I should add mouse CD3, so mouse version of teplizumab. Both of these saw incredible synergy where we were able to reverse these mice durably almost 90% of the time. And so with that, we said, aha, we can do a trial with ATG and GCSF in humans, 2 FDA-approved drugs commonly used on the market for a while. We can scuttle the Cytoxan, and maybe we'll have a therapy that can have some legs. And so that led to a Helmsley Trust-funded pilot trial, where we were really trying to push the paradigm to suggest that we didn't need to follow the old mantra that type 1 is an immunologic emergency, such that if we don't do anything within the first 100 days, all is lost. And so we intentionally designed the study to be in folks who were at least 4 months post-diagnosis, so we didn't compete with new onset trials, but out to as much as 2 years post-diagnosis. And we used very low dose thymoglobulin, so 2.5 milligrams per kilo, which is about the lowest dose you can find in the literature used for aplastic anemia. We used a standard course of Neulasta, 6 milligrams every 2 weeks. And we did a 2-to-1 randomization with drug or placebo. And this initial study was single-blinded. And what we saw was that even in a really small trial, a year post-therapy, the average C-peptide in those who got combo was actually above baseline and significantly better than placebo here in the light gray. And even as you follow these patients out to 2 years, while you lose statistical significance, again, because the sample size is so small, you can appreciate that there was still a difference in C-peptide production in those who got the combo therapy versus placebo. But of course, this was a small pilot, and we wanted to see if this was a real observation. And so we then were lucky enough to take this approach to TrialNet. And TrialNet agreed to let us do a confirmatory study in new onset patients with the goal of confirming the safety and efficacy of this approach, but we wanted to answer more than just one question. I should get all credit where it's due here. I initially said, let's just repeat the study and do the simple, kitchen sink ATG GCSF versus placebo. But better heads prevailed and said, well, you really haven't answered the question as to whether or not low dose ATG by itself is driving the benefit. We knew by this point that high dose ATG from the START trial actually didn't work. So the question was, is it the low dose ATG plus the GCSF really, or is it just the lower dose of ATG that's driving the benefits? So we smartly designed this as a 3-arm trial to ask combination ATG alone versus placebo in new onset type 1s. The therapy was exactly the same as what we gave in the pilot study. And we used the typical new onset definition, again, going back sort of to the 100-day arbitrary definition. And we enrolled folks between 12 and 45. The primary outcome for that study was, like most, a 2-hour mixed meal tolerance area under the curve C-peptide at the 1-year endpoint. Although we did collect 4-hour mixed meal tolerance test data on these patients as well. They showed the same outcome data. We again wanted to make this relatively simple, and so we decided we'd stick with a 2-hour study. And we needed 84 subjects to have the appropriate power. One important thing to keep in mind, which you'll see in a second, is that we had a 3-arm trial here. And so in order to detect a 50% increase in either experimental groups, we actually had to accept an alpha of 0.025 for significance instead of your typical 0.05 if it was a 2-arm study. And you'll see why that matters in a moment. Before I get to the end results of the study, I'm always proud to show this. I just think PROUNET does an incredibly good job of running clinical trials. And you can see that we screened 113 subjects, 89 were randomized, 29 and 31 were in the 3 groups. But even at a year, we had 87 of the original 89 folks randomized finish follow-up. I think that's really impressive for a trial that required as much intervention as this did. And even at 2 years, we had 82 of the 89 follow-up, so very little dropout. In fact, most of the dropouts that we had were literally right at randomization. So one subject withdrew, they got randomized and never even got drugged. And one got drugged and then withdrew from the study. So literally everybody else completed follow-up at least through 1 year, which again is very impressive, I think. All right, so let me show you the results. And I'm going to skip forward just to show you the 2-year results because we now have those data in hand, and these were published last, now almost a year and a half ago in Diabetes. But somewhat to our surprise, when we looked at the data, what we saw was that low-dose ATG by itself, not in combination, actually provided the most significant difference versus placebo. You can see with a highly significant p-value here. And while ATG GCSF seemed to be in between the two, and the p-value was, and you normally think of less than 0.05 as significant, remember we had to have that alpha of 0.025 because the study is designed to have power. So this combination was actually not significantly different from placebo, although when you look at the absolute values, you can appreciate what's going on here. And what's more challenging to describe to most folks is that because of the way we set up the power analysis, we weren't able to statistically say that one of these therapies was better than the other unless both had demonstrated significance versus placebo. So while, without statistics, you can say, obviously, well, ATG alone outperforms the combination. Formally and biostatistically, we can't quite say that. So all we can really say is that low-dose ATG is clearly better than placebo, and the combination is not. But at 2 years, again, this is a pretty impressive finding, one that we haven't seen in many other interventions in the type 1 space. And beyond that, patients want to know, well, how does this apply to my A1C? C-peptides all good and well for you scientist folks, but I want to know what this does to my control. And here what we saw, and again, this represents sort of the overlap of the 2 agents, is that the low-dose ATG and even the combo were both highly significant in terms of reducing A1C compared to folks who got placebo. That's not to say that A1C didn't do what it always does, which is nadir right after honeymoon is in full effect and then still go up. So again, while we are seeing benefits from these approaches, we've got a lot of work to do to keep the A1C down and maintain it in that space. Adverse events, of course, were an issue and remain an issue for any of these drugs that have been therapeutic. I think that's the big challenge with type 1 is deciding what you're comfortable with in terms of risk-benefit. The primary side effect associated with anti-thymocyte globulin is cytokine release syndrome and serum sickness. And almost everybody who got active drug had some degree of cytokine release. although that's seconds to minutes and during the infusion only and doesn't recur. And the majority of them, good 2/3 to almost 3/4, also had serum sickness, which is a good 2 to 3 days of feeling pretty darn miserable. Hand and feet swelling, lovely rash, some nausea, malaise. So I imagine it feels akin to what some people feel when they do have COVID. We always describe the patients as feeling like they have the real flu. So not a complete walk in the park, but certainly self-limited, well-known, well-described, and easily managed, but not something that we would easily compare to placebo in terms of the side effect profile. And more importantly, as we move on from what did this drug do for our patients and how did they tolerate it, we wanted to understand how the heck it worked. And unfortunately, we still don't completely understand. But this is a work very much in progress with many of my colleagues at the University of Florida and internationally, even to understand how low-dose ATG affects many pathways. I think one of the challenges of it being a biologic agent instead of a pure monoclonal is that it does so many things, it's hard to be entirely sure. But to summarize it, at least when we looked at the 6-month flow data after drug, it resulted in a moderate increase in APCs, but very, very, very quickly. We saw, unlike high-dose ATG that actually preserved CD8 Tregs and CD8s total, we saw that the CD4 Treg count went down only very minimally and just very quickly, and then was preserved with increased memory. Again, that's very different than we see with high-dose or higher dose ATG, like the dose used in the START trial. And then we did see that the CD4 conventional cells were depleted, naive more than memory, but that they do sort of come back to baseline over a period of 6 to 9 months, with PD-1 positive cells persisting, and some evidence that we might be inducing some degree of exhaustion, which of course would be exciting for providing some sort of long-term benefit. And so to summarize kind of where we are with the ATG data, the low-dose ATG clearly preserves C-peptide in new onsets all the way out to 2 years. The combo does not. Again, so it doesn't outperform it. So for practical purposes, you would say, oh, let's just drop GCSF for future studies, and that's our plan. That doesn't mean we're abandoning combination therapies at all. It means this just isn't the one to combine with low-dose ATG, and we should pick some other drug. It does have clinically meaningful endpoints like reduction of A1C out to 2 years. I didn't show it, but I should note it did not have a meaningful effect on insulin requirements, although I'll always argue that I think insulin reporting in terms of dosing is highly variable with patients using different lifestyles, low carb, high carb meals, et cetera. So I don't know what that data really means. And there's some nice mechanistic data to support the pathways where this is working. So let me pause there and suggest let's shift gears a bit. So, I focused on ATG because that's the trial that our group here has sort of been most interested in the last decade. And say, I'm actually an optimist most of the time. So let's be half glass full and say, well, now we have all these drugs, at least a handful of them that have shown benefit. We should be asking of those, which ones perform the best. And certainly the best way to do that would be a head-to-head clinical trial where you have 6 or 7 groups and you're randomized to all the different agents. Unfortunately, that's not likely to get done anytime soon, if ever. And so our group decided that we should try and do a cross-trial comparison to do as close of an apples-to-apples look at which drugs outperform others, fully appreciating that you have to take a grain of salt with it and that you can't really do a true apples-to-apples comparison. So I won't bore you with the statistical details on the slide, but I'll skip to the next one. And so we just decided to do a comparison of the new onset studies that we could get publicly available data for which there was a positive endpoint. So again, the abatacept study in TrialNet, rituximab in TrialNet, the low-dose ATG study that we did in TrialNet, alefacept, which again is not available on the market, but really had some impressive outcomes in the TIDAL study run by the Immune Tolerance Network. And then the teplizumab study, and I don't mean here the recent data from the prevention study, but this was the ABATE trial, which was a new onset study. And again, there have been a number of teplizumab studies. So this is a limited look at one of the new onset studies, but the one whereby we could get data easily available to us publicly. And then we threw into our analysis the high-dose ATG study, just for comparative of something that didn't work, so that we could see what that looked like graphically. And so again, I won't bore you with the details of how this analysis works, but just to orient you, we did this 2-year trial comparison. So looking at the treatment groups 2 years out from these 6 selected studies, and we looked at the C-peptide AUC mean. And what we're looking at are in the black dots, the placebo group, and then the triangle is the experimental group. And the best way, per the statisticians, to describe these data are that the difference between the gap between placebo and experimental group, regardless of where it falls on the figure, actually gives you the most information about which study performed better than the others when you compare across them. And so, of course, the first critique people always say is, well, the placebo groups acted differently, so how can you do this? Yes, we know that. And while graphically this is how these are shown, this is still by far the most objective way to try and assess is one intervention better than the other. And what we see is very clearly low-dose ATG and teplizumab. I'm sorry if those red lines are covering up their names. This is low-dose ATG here, and this is placebo. And this is teplizumab. At 2 years post-treatment in new onsets, this is the difference between the effect compared to rituximab as a baseline for those. So 300% better than rituximab and 200% better than rituximab for ATG and teplizumab, respectively, and clearly outperforming all the other new onset agents. I know this is a little bit of a contrived analysis, but I still think this is really important data and stuff that we should be considering as we talk to patients, families, and researchers as to what should we do next. Especially in a time of COVID, when you're saying, all right, what therapies are most likely to provide benefit if we're going to think about doing trials now or in the future, I think you want to know which ones work the best. And of the data we have available to us now, it's absolutely these two. It doesn't mean the other ones shouldn't be included in some combination approaches down the road or be considered as personalized medicine for folks who might be best suited for the drugs that affect particular pathways for them. But there's no denying that these two agents work quite well in the new onset space. All right, so that's led us to where do we go next with ATG? And frankly, if it hadn't been for the pandemic, we would already be doing this study. But TrialNet's model is very much to do new onset studies and then try and move them into as proof of concept, and then move them into our population of most interest, which are high risk antibody positive folks who we think are going to progress to type 1, and try to see if we can prevent the disease. So we designed a low dose ATG monotherapy, ironically, prevention study aimed at stage 2 type 1 patients. So these would be antibody positive folks with dysglycemia. We wanted to move the age down to enfranchise more of our patients who get screened in TrialNet anyway, who are high risk. So we went to 6 and up because this drug is used in patients 2 years of age and older on a regular basis. We were going to stick with a single course of low-dose ATG, so just the 2.5 milligrams per kilogram, which I didn't talk about how it's given, but it's a 2-day outpatient IV infusion. So it's actually practically also has some advantages over teplizumab, which requires 12 days of visiting an infusion center in a row. There's the two IV infusions. This study would be 1-to-1 randomized, again, just to help us out statistically, because it still requires 97 subjects to have enough power. Now, it's important here to think about just how darn hard it is to do prevention studies in the type 1 space, right? If we were to enroll this study at historically sort of low rates for picking up these groups, it would have required 6.5 years to just enroll the darn thing, assuming you were able to get 15 folks in per year. It'd take you almost a decade to answer. That's not acceptable to me and most folks in our field anymore. As good as the teplizumab data are, and they did take 10 years to develop, I don't think we can tolerate getting one answer every decade. So we really have to think about other designs. And so the questions come about is, well, what if we start designing studies to not be so line in the sand about what a diagnosis of type 1 looks like? Why can't we accept that it's really a continuum and design our studies to go fully across stage 1 and stage 2 and stage 3? We could make the studies based on C-peptide outcomes and no longer be glucose-centric. When my glucose is above 126 and I've met threshold, is that really as important as whether or not my C-peptide has declined or maintained stability? And we could have mixed eligibility. So new onsets would be those folks who are stage 3, as well as folks who are confirmed abnormal glucose tolerance, those folks who have stage 2. You could stratify to ensure balance. And this would allow evaluation of multiple experimental regimens versus an overall placebo group. And so just as a schematic, here's one way to design a study like this. And I really do think this is the way we need to be moving, because I think this gets us closer to my title, which is personalized medicine for type 1 prevention. Again, we got to get away from this one drug versus one placebo and moving on. So if we have a study designed where we have multiple opportunities for re-randomization, and here just for illustrative purposes, this is placebo versus rituximab versus ATG, a sort of induction versus placebo, and then you would re-randomize to get a second agent. Here it's Rituxan and abatacept, here it's ATG and abatacept. You get to answer 4 or 5 different questions with a 2 to 2 to 2 to 2 to 1 allocation randomization. This is really far more efficient for us as a society than doing these one-off trials. It does require a bigger commitment up front of both patients and the community agreeing to it, and we have to be very thoughtful about how we pick our agents. But there's no doubt that over the same period of time, if we had designed studies like this, as opposed to the way we've done in the past, we'll be able to get closer to our ultimate goal of therapies that A, work, and B, we can translate into therapies for patients. So in addition to that, we've always just randomized folks based on, yep, they got risk for type 1 and, we have this drug. But what if we were able to use a priori some understanding of their likelihood of response? And this is getting into the idea of personalized medicine. Everybody is very hot right now with these ideas of trying to understand when we look at clinical trials. Sure, it may have worked for the large group of folks who got therapy, but we always know that there are individuals who get active treatment who get no benefit at all. And there are other individuals who get active treatment who are driving a lot of the response because they're what we might call super responders. And so I asked this almost rhetorically, but should we be including these other measures of likely response, assuming we can understand them? How does HLA play into this? Should we be doing full exome sequencing to understand the specific pathways most likely to be driving autoimmunity in a type 1 patient? Should we be looking at specific flow data, functional data with T cells and B cell responses? Should we be looking at other things outside the box, like pancreas volume size? So we have some nice data that I'll show with you in a minute, that the pancreas volume is markedly reduced in type 1 and in pre-type 1. So maybe if you're a multiple antibody positive person with a high-risk HLA and a big pancreas, your time to diabetes is going to be much longer than somebody who has the same other risk factors, but a much smaller pancreas. And so should we be using all of that to put people into study groups and stratify them a priori? I think the answer is yes. I think the challenge is we still don't know quite which ways to do that just yet. So getting back to the prevention study with ATG, the TrialNet team sort of went back to the bullpen and said, all right, guys, we need the statisticians to help us come up with ways to do this in a more efficient way. And so what we did is we used some more fancy risk estimation to reduce the sample size needs. Starting with first the idea of if you took multiple antibody patients who were ever multiple antibody positive with no confirmatory data and no ambulatory, abnormal glucose tolerance confirmation required, we identify almost 100 of them a year. And we can start to identify their 2-year risk for progression to type 1. So a really narrow time period where their risk would be really high. And so as we worked through this, we got down to, we can see lesser numbers for different versions of risk. But by doing this and focusing on folks who are going to progress from not just stage 2, but from stage 2 to stage 3 in only a 2-year period, using this column here, we could really start to talk about, all right, these are going to be the most high-risk patients. We can, A, justify applying the most aggressive therapies to them, and B, it allows us to shrink our sample size because we're going to have more events. And the challenge with the teplizumab prevention study is we had to just wait so darn long for enough people to, always sounds bad to say it that way, but for people to develop type 1 to know if the therapy was effective or not. So designing trials where we're focusing on this even more high-risk group will allow us to enrich the study and get to an endpoint sooner. All right, so back to the idea of pancreas volume. I just want to mention this briefly because Columbia is going to be a site helping us with recruiting for the study, so I wanted you all to know about these data. We've known for quite a long time that the pancreatic mass in patients with type 1 is lower than people without diabetes. And then a few years back, the NPOD team based here at UF did some work that confirmed this, where they looked at the weight of organ donors who died with type 1 and those who didn't have type 1, and again confirmed, yes, people with type 1 have less pancreatic weight than folks who don't have type 1. But what was really interesting was that when we looked at those who just had autoantibodies, which are these rare patients that we do get from the organ procurement organizations through NPOD, they were in fact in the middle. So folks who had antibodies but no dysglycemia, no history of diabetes, did have reduced pancreatic mass compared to true controls, but not as low as type 1s. And so that really led us to think, could we use non-invasive measures of pancreas volume as another way to stratify patients? We were fortunate enough to get a DP3 NIH grant to look at this cross-sectionally. And long story short, we found that, yes, in fact, if you go from control patients here in green to antibody negative first-degree relatives, so even folks who don't have a single antibody, but they just have a genetic association by virtue of being a relative, their pancreas volume is already reduced. I think that's fascinating. And then you go to single antibodies and multiple antibodies, and you get yet further reduction. And then, of course, you go to the type 1s in blue, and their pancreas is often 50% reduced in volume compared to folks who are controls. And so, the challenge here is that this is all just cross-sectional data, and we really want to understand what's the natural history of this decline. And so we recently applied for and were awarded a new R01 with Martha Campbell Thompson and I as the PIs. And we're going to run this as a TrialNet ancillary study. And the study design is really quite simple, but we'll need 144 subjects. And the idea here is that we're over time going to be able to develop a personalized risk score by using pancreatic imaging. And it's just going to be a simple clinical style MRI done at baseline and a year later. And we're going to do some endocrine functioning, of course, like we always would do in TrialNet, so glucose tolerance testing and CGM wears. And then we're going to look at exocrine pancreatic function, because we've recently, in the last couple of years, been able to further document that while not clinically relevant, exocrine function is in fact reduced in most type 1 patients. And then we're going to look at all the associations with the genotype. And so we need a good number of patients to enroll in the study, and we're excited that Dr. Golan's agreed to be a site for this, and so you guys will hopefully be enrolling folks. It's as simple as a family wants to be a participant in TrialNet and get their antibody status known. They would come in and be screened. Their antibody status would put them into one of 3 categories for this trial, which would be antibody negative, a single antibody positive, or multiple antibody positive. Those are our 3 groups. And then they would have the MRI and metabolic testing done at baseline, and then 1 year later. So a really simple study design, but one that we hope will answer a lot of questions as to how does that pancreas volume reduction progress over time? What is the natural history of that? All right, and then moving on to another topic that I think we can discuss as a group perhaps is, given the elephant of the room that is COVID-19, how do we move forward with clinical trials right now in the type 1 space? Obviously, doing a pancreas volume MRI is low risk, other than having to be around somebody. But I just spent a bunch of time telling you about all these immunotherapies that we think, quote unquote, work, but they clearly, cause lymphopenia and increase potential risk for, secondary exposure infections. How do we help patients, families, clinical trialists designing studies decide what's acceptable? So our team put together a perspectives piece that's under review right now at Diabetes. And one of the things we put in it was this theoretical construct about relative risk versus likelihood of benefit. And, should this really matter in a time of deciding whether or not you want to do a trial when you might get exposed to something like COVID-19 or frankly anything? I'm probably a little bit more risk tolerant than the average person in the type 1 space. But it's funny, when you show a curve like this to people who are treating other autoimmune diseases that aren't necessarily worse than type 1, things like rheumatoid arthritis, or JIA, or even inflammatory bowel disease, they consider these drugs up here at the top of our risk tolerance curve to be standard of care already. And you might argue that's because they have data that they work, but we can't ever generate those data if we're not willing to do the trials. And in fact, the drugs up here at the high end of the risk curve are the ones that have provided evidence of benefit to date, whereas the ones down here at the lower end of the risk curve are the ones that have uniformly not demonstrated benefit. So I show this, again, as a bit of a thought experiment, but one I think is going to have to actually drive our decision-making as we move forward in the type 1 space. Which studies are we willing to do? How much risk are we willing to take? And how can we mitigate against those risks so that we can move forward? Because I don't think it's reasonable to say we're just going to stop completely. And we did that with our type 1 trials earlier in the year when we didn't know enough about COVID, and that point was entirely reasonable. But we can't do that forever, and we have to figure out a way to move forward, appreciating that unfortunately we're all going to be living under the cloud of COVID for at least the next 12 to 18 months as vaccinations roll out. But our kids are going to be the last to get those vaccines, and many of them are the ones who could benefit from these studies. All right, and then last thought experiment I want you to do is think about cost effectiveness, right? We haven't been able to have this discussion previously in the type 1 space because frankly, nothing's worked well enough to even really consider it, but we're there now, okay? Teplizumab is literally on the cusp of getting an FDA label for preventing type 1 diabetes. The FDA has given it its fast track course. They might even get a green light by as soon as July of this year. That's really quite amazing. But to me, one of the elephants in the room is that the company and other medical economists have said, they're probably going to want to collect a six-figure ransom for this drug. And I'm not saying that because I have any problem with people in medicine making a profit, or that they got to get all back the R&D costs of it. But that's not a trivial amount of money, of course. And so the real question becomes, well, what is the cost effectiveness of that therapy? And I think insurance companies are going to pay a lot of attention to this when it comes time to decide, all right, fine, the FDA gave you a label, but are we going to pay? So another example of a drug that's just recently worked is golimumab, right? So golimumab is not a cheap monoclonal antibody. It's about $5,000 a dose. If you're talking about using it continuously, like they did in the Tiger study that showed beautifully that it preserves beta cell function, you're talking about $130K retail a year, right? That, again, is not a trivial cost. Now, these drugs are pretty darn clean and have pretty minimal side effect profiles. Teplizumab still has some cytokine release, but no serum sickness. Golimumab, almost nothing. They both certainly cause their relative degrees of immunosuppression. But again, can we justify the cost effectiveness there? And I point this out just to compare to sort of the drug that I've been working with the most lately, which is the anti-thymocyte globulin. Anti-thymocyte globulin is dirt cheap relative to these. I mean, that kind of money is certainly a lot, but you're talking an order of magnitude less per course of drug. And so I've been fortunate enough to have a number of families come and request clinical treatment now with ATG based on our previous studies. And at first I said, we can't do that. We don't have enough data to support that. But now, having finished 2 clinical trials with good results, I finally sort of agreed to treat a family who, dad was a medical professional, mom had type 1. They knew exactly what they were getting into. And I said, all right, well, this is going to be expensive probably. And lo and behold, the insurance companies have to date been willing to cover this, with the patients only covering their copays, which for most of our patients so far we've done this on has been less than a couple $1,000s. So again, I think it all just depends on where we're going with this. And so we were fortunate enough to get contacted by a medical economist at Memorial University up in Canada, who focuses on these kinds of issues. And he's helped us do an analysis that is still being finalized right now. But the one line kind of bullet point for now is that, even if ATG as a comparator is 50% less effective than teplizumab, if teplizumab is 66 grand, which at this point looks like it's going to be considerably more than that, ATG is still far more cost effective. And so I'm not going to show you all the background data to show that up, but I just think that's an interesting thing that we should be thinking of as we're fortunate enough to move into an era where we have therapies that actually work. And again, work in quotations is what that means for individual patients, more so than what it means because of a p-value in a group. All right, so lastly, and then I can answer some questions. I think these are the real questions that are in front of us as clinical trialists in the type 1 space: is what the heck do we do next, and how do we design the trials to make them most efficient? I certainly still believe very firmly in combination approaches. And so I think we need to be choosing them wisely. Should those drugs be chosen based on age? Do we have a biomarker we believe in that can help us choose which patients gets which one? Should we really be focusing on prevention or new onsets? I think is a fair question. Do we use any of these drugs as induction therapy? And what the heck do we do about retreatment? I mean, to date, teplizumab is really the only therapy in a currently available new onset trial that has visited the idea of treating a patient again, which is unlike any other autoimmune disease, right? You don't treat somebody with JIA once and walk away and say, have a nice life. We treat them, or IBD or any other disease for that matter. So I think we have to start designing trials that do that once we have a reasonable cadre of drugs that provide benefit. And I think we're there. Do you want to just consider ongoing active therapy with a drug like golimumab that's short acting, but clearly has had some benefit? And I didn't even mention the other elephant in the room, which is that Janssen has decided that they're not particularly interested in pursuing this drug, not because it didn't work, but because of the economics of recovering, a profit on it. I think that's the sad state of affairs, but it's just the reality of our field, and we have to acknowledge that. And hopefully somebody will produce it as a biosimilar, and they'll still be interested in it. Do we still envision the use of antigen-specific therapies? We've never really given any of these under the cover of an immunomodulating, immunosuppressive induction therapy. I think there is some benefit potentially there, but we don't know. We haven't answered that question, and I think we need to. And then any of the other drugs that are out there. There's so many other combinations to consider, but I think we need to be considering them all holistically when we think about how to design our trials. And I will end there and just give thanks to all the folks who make these studies possible. Certainly the Helmsley Trust for taking a leap of faith with us back when we did the initial ATG-GCSF combo pilot. TrialNet for letting us move the ATG monotherapy study forward. Sanofi for providing us a drug for the original pilot. Amgen for providing the GCSF, and the NIH and the GDF for always being our hugest supporters. As with anything in science, this is an incredible team effort, and this is just a small smattering of the folks here at the UF Diabetes Institute that make all the magic happen. And so, anything I said today was on behalf of all of these folks who do incredible work, and perhaps much more importantly, on behalf of these kids. Again, I'm a pediatric endocrinologist, and you guys see adults too, but this is who we should be focused on when we're thinking about designing trials. How can we develop therapies that are going to be meaningfully beneficial for these kids as fast as we can get them into them, so they no longer have to live a life affected by type 1. And with that, I will stop talking and see what questions you guys have.
Well, Mike, thank you. That was terrific, very thought-provoking. Mimo, you're clapping. Did you have a question?
No, I just have a comment that I really enjoyed this. It was very thoughtful and thought-provoking, and I really appreciate, the efforts you're making. Very well done.
Thanks, Mio. Appreciate it.
So, Mike, if the FDA approves teplizumab, how would you envision our treatment of people with new onset type 1 diabetes to change?
Yeah, so their initial label is going to be, if they get it soon, will be in prevention, which I think is interesting. Not technically a new onset, but we all know things happen once a drug is labeled at all by the FDA and people will seek to use it off label. I think a fair percentage of patients, are going to very quickly start demanding this as an opportunity. I don't think it's going to be all of them. I think it's actually going to be a pretty small minority at first, but that will grow with time, assuming that insurance companies pay for it. And once that becomes any sort of standard of care option, I wouldn't say standard care, then I think we have to start designing our clinical trials with that in mind as well. And there was just a nice New England Journal piece today about, maybe it was yesterday, but about COVID-19 vaccine studies and placebo being so necessary to get the answers. But once you have an active therapy that's demonstrated to work, it's really hard to get people to do a study that's placebo controlled. And so we might have to start designing our trials to have an active comparator arm almost always. And beyond being hard to do, I think it becomes ethically challenging to suggest that you're not going to offer somebody active therapy if you really believe it works. Comparing it to COVID-19 vaccine may be a little bit of a stretch, but I don't think it's that far off.
Questions? All right, I have another question. Why is the pancreas smaller?
Yeah. I mean, like everything in type 1, multiple factors, right? I can wave my hands. There's clearly some genetic component to that based on what we saw from the FDR, pancreas volume and mass data from NPOD and then from TrialNet. So I think there are going to be HLA and SNP drivers of that. So I think you start with a smaller pancreas, and folks who are probably most likely to get type 1 at an early age. That's a hypothesis that we need to try and see if that's true with the longitudinal study. And then I think we've just long ignored the exocrine pancreas as being part of what happens in type 1, and it clearly is involved. It may be a bystander effect, but the immune response is maybe not so purely affecting just beta cell mass. Obviously, it affects function for lots of other things within the pancreas as a whole. But hypotheses from something as simple as the loss of local high exposure to insulin as a growth factor to the inflammatory response. Are T cells really invading as they get into the pancreas, or are they actually attacking exocrine tissues as well? So we're going to be looking at those things in NPOT as well. So doing live slice tissue studies to see if we can see really the pathways that are going on to explain why the pancreas is so much smaller, because it's not trivial. It's almost a 50% reduction in volume and mass.
So in rodents, when you knock out the insulin genes or the IGF-2 gene, the exocrine pancreas is much smaller than any other organ in the body.
Yeah.
Consistent with what you've just said about the trophic role of insulin and IGFs on the exocrine pancreas. It is interesting, though, that this seems to precede the actual onset of diabetes, suggesting that maybe the underlying pathophysiology has been brewing for longer than we thought.
I agree.
Hi, Dr. Hiller. This is Lee. A great talk. I'm a basic scientist. I have a very naive question regarding the type 1 diabetes pathogenesis. So those beta cells should be attacked by some autoantibodies or something, and then they'll be killed. So my question or imagination is there a way we can hide beta cell from those autoantibodies? Or in another way, if the autoantibodies recognize anything on beta cell, then the beta cell destroy these autoantibodies. Is this a potential way to treat or prevent type 1 diabetes?
Yeah, so the autoantibodies in type 1 diabetes are not pathogenic. They are sort of the smoke of a fire. So they're just evidence that there's an immune response ongoing, which is, we believe, primarily T cell driven. So you can remove the autoantibodies and it doesn't actually change the immune response per se. You can adaptively transfer autoantibodies, and that doesn't confer disease. But if you adaptively transfer autoactive T cells, it sure as heck will cause the diabetes in animal models primarily. So I think all the drugs we're talking about essentially do that, and they're trying to disrupt that normal, the abnormal pathways there that are leading to type 1 by either blocking the T cells from getting to their final target, reducing them entirely, like immunodepleting agents like teplizumab and ATG, but also some of the more complex things, which I breezed through at the beginning. But even B cell agents, which we used to think of like this is a T cell mediated disease. What is anti-CD20 going to do? It's a B cell agent, but when you deplete B cells, and B cells have to essentially educate T cells to know what to do, you diminish the T cell's activity, and you do in fact provide some benefit. So hiding the beta cells from the B or T cell activation, I think, is key. Pie in the sky would be to really induce tolerance. And so if you induce tolerance because you expose the immune system to an antigen that it decided it liked as opposed to disliked, then I would think the antibody levels would over time diminish, and you'd still preserve beta cell function. We have yet to be able to do that in the type 1 space that I'm aware of.
Okay. Thank you.
Okay, any last questions? Michael, thank you so much for coming and for giving this terrific talk. And we will see you soon in person or virtually. Thanks again.
Sounds great. Thank you, everybody. I appreciate it. Have a great day.